चलिए रिवाइज कीजिए जेनेटिक कोड 6.6 जेनेटिक कोड के बारे में रिवाइज कीजिए फटाक से। रेप्लिकेशन एंड ट्रांसक्रिप्शन के दौरान ए न्यूक्लिक एसिड वाज कॉपीड टू फॉर्म another dash dash, hence these processes are very easy to conceptualize on the basis of dash dash. The process of translation requires transfer of genetic information from a polymer of dash dash to synthesize a polymer of amino acids. Neither does any dash dash exist between nucleotides and amino acids, nor could any be drawn theoretically. There excited ample evidences, though, to support the notion that change in dash dash acids, that is genetic material, were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of dash dash acids during synthesis of proteins. In determining the biochemical nature of genetic material and the structure of DNA was very exciting. The proposition of deciphering deciphering of dash dash code were most challenging. In a very true sense, it required involvement of scientists from several disciplines. Dash dash, comma dash dash, comma dash dash, and a dash dash. It was dash dash a physicist who argued that since there are only dash dash bases, and if they have to code for dash dash acids, the code should constitute a combination of bases. He suggested that in order to code for all the dash dash acids, the code should be made up of dash dash nucleotides. This was a very bold proposition because a permutation combination of dash dash would generate dash dash codons, generating many more codons than required. Providing proof that the codon was a dash dash was a more daunting task. The dash dash method developed by dash dash was instrumental in synthesizing dash dash molecules with defined combinations of bases, that is dash dash and dash dash. Dash dash cell-free system for protein synthesis finally helped the dash dash to be dash dash. Dash dash was also helpful in polymerizing RNA with defined sequences in a template independent manner, that is dash dash. Finally, a checkerboard for genetic code was prepared, which is given in table 6.1. माइंड कैप्चर कीजिए और बनाने की कोशिश मत कीजिए बल्कि बना कर दिखाइए इस टेबल को मिनिमम 20 बार। 6.1 द कोडॉन्स फॉर वेरियस एमाइनो एसिड्स। बना कर दिखाइए जल्दी से। तो साइलेंट फीचर बताइए जेनेटिक कोड का। चलिए फर्स्ट पॉइंट, द कोडॉन इज ट्रिपलेट डैश डैश कोडॉन्स कोड फॉर amino acids and dash dash codons do not code for any amino acid, hence they function as stop codons. सेकेंड बोलिए। जी हाँ, कुछ अमाइनो एसिड्स कोडेड होते हैं बाय मोर देन वन कोडॉन, इसलिए द कोड इज डैश डैश। थर्ड पॉइंट बोलिए। The codon is read in dash dash in a contiguous fashion. There are no dash dash. फोर्थ पॉइंट बोलिए। The code is nearly dash dash. For example, from bacteria to human, dash dash would code for dash dash. Some exceptions to this rule have been found in dash dash codons and in some dash dash. फिफ्थ पॉइंट बोलिए। Dash dash has dual functions. It codes for dash dash and it also acts as dash dash codon. सिक्स पॉइंट बोलिए। डैश डैश comma dash dash comma dash dash are stop terminators codon. If following is the sequence of dash dash in mRNA, predict the sequence of amino acid coded by it. Take help of the checkerboard. चलिए सॉल्यूशन कर लिए। नेक्स्ट, नाउ ट्राई द अपोजिट। फॉलोइंग is the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? 6.6.1 Mutations and genetic code we will discuss in the next session.